Hello, Anupama. We should start, I think. Is, is
Ma'am,
Shweta control?
students are joining in. We'll wait for 2 minutes.
There is some echo. Please look into that.
Yes.
Did you have a class just now?
They are just finishing with their practicals and joining them.
Actually, I can't hear whoever was speaking just now. Can you just speak up just in case the voice is not loud enough? Susan, maybe you just say hello so that I know
Hello.
I
Yeah, I
can.
can hear you.
Okay.
Suraj, I can hear you. But
Okay.
there is some background. I think some mobile is nearby. Yeah. Some mobile
Just
is nearby. Yeah.
yeah.
Somebody's mobile is making a sound.
Actually, I moved mine on the chair and away. I just kept it on because sometimes when we are muted, if some message is passed on, then you don't know about it. Otherwise, I'll just switch it off. Yeah.
Yes. It's not in silent, but reduce the volume.
And shall we start? Hello.
Yes.
Shall we start, ma'am?
Yes, please. Yes.
Good morning and a good morning and a warm welcome to all. Myself, Shweta Patel, on behalf of the Department of Microbiology and Biotechnology of Patkar-Varde College, warmly welcomes you all for the webinar on scientific writing. Before introducing to our speaker, I would like to request Dr. Sureka Jinde Ma'am, Member Board of Trustees, IWSA, Indian Women Scientist Association, to tell us about IWSA.
Thank you, Shweta.
Thank you, ma'am.
Good morning to everybody. Uh, I'm Dr. Sureka Zingre, trustee from Indian Women Scientists Association. On behalf of my colleagues and chairperson of the Board of Trustees, members on the executive committee, as well as other members from IBSA, we welcome you all to this meeting, which we are now having a lecture from Dr. Susan Nippen. I'll just share the screen with you to see our activities before we move on and leave the stage for Dr. Susan so that you have an idea as to what we are doing and how we do things. Can you see that loading?
Yes.
Is it seen?
It is coming, but not come fully.
You'll have to share the PowerPoint, ma'am.
Yeah, sorry. Now is it there?
Yes.
Okay. So can everybody see it? Suparna, is it clear?
Yes, ma'am.
Yes, ma'am. Full screen.
So on behalf of all of us, we welcome all of the participants for this program. We are the Indian Women Scientists Association, and an organization which was started way back in 1973. We are now in our 49th year, moving ahead to face the 50th year, good golden jubilee that we are looking forward to. The picture that you see right in front of you, with my pointer I'm showing you, is our headquarters at Vashi, and this is a hostel that we have there and other activity centers which I'm showing here, along with a garden. We are registered with the public trusts as well as Niti Aayog, which is a requirement for all associations and NGOs. IBSA is a non-political, secular social welfare organization. As mentioned, in its 49th year, it was started by 12 scientists, well acknowledged scientists who believe that science should not remain in the four walls of the laboratory but should move out to society. And over the last 49 years, besides the headquarters in Vashi, they established the 11 branches across the country, which are Roorkee in the north and Bangalore and Nellore in the south, and the rest of them you can see out here. Our main mandates is to take science to society and for women empowerment. And we do this through two arms via community welfare and science awareness. During 1920, 2019-20, we reached out to almost 23,000 recipients. And during 2021, we didn't get left behind because of the pandemic. We could reach out to almost 17,000 individuals who could benefit from our outreach programs. Under the community welfare activities, we have a working women's hostel which was established way back in 1989 on the Vashi campus, a space for 160 working women who may earn anywhere between 10,000 to 50,000. And it's a clean, efficient, economical place working under the guidelines of the Women and Child Development of Maharashtra. So we encourage young girls who are looking for a place to stay, do consider looking at the Working Women's Hostel here in Vashi. And you can, it's very close to the railway station, so you can actually get back to college also if it's required. We have a healthcare center which was started way back in 94. We have a physiotherapist and a dentist, and we run different camps. We have a computer center which has completed 30 years. We have programs for little children up to senior citizens. This is the digital world, so we all have to know about computers and how to use them, and use them very well and efficiently. A library which was started almost with the inception of the association and now is hosted and housed in the Vashi campus. We get support from the Maharashtra State Libraries, and we have around 5,000 books. We have a scholarship awards wherein we provide girls who are in the 10th, the 12th, the BSc, MSc, and PhD one-time scholarship award for meritorious studies that they are undertaking. And this is possible because of the donations received from well-wishers to IVSA. And the interest earned from those donations is what is given to the youngsters. And here we just completed this year's award session with Dr. Vasudha Kamath, the past Vice Chancellor of SNDT University. We will be having that again this year. So I encourage young girls who need some support to please log on to our website and see when these awards are given and when you can apply. We have what we call as the Early Childhood Care and Education Program. I will tell you a little more in detail later. A nursery school and a daycare center. During this time, the daycare center has been temporarily closed and we'll be opening it again now that the pandemic it is under control. The nursery school has started recently, and it was way back in 93. These two programs go hand in hand with the ECC program. So as mentioned, the ECC program is a one-year diploma course affiliated to the SNDT Women's University. Any person who has completed that 12th standard, any age is no bar, can undertake this program, and then they become eligible for teaching in daycare and nursery schools. It's a wonderful vocational program, and every student which passes out from this gets a job. We can almost guarantee you that. Sometimes we have people asking for students even before the academic year is beginning. We work hand in hand with Drishti, wherein we have a shadow teachers training program and a program to understand learning deficiency for children so that the whole teaching exercise is inclusive. We've joined hands also with Vigyan Prasar and we've run a course with them where the course was conducted by the ECC teachers for the pre-primary and primary teachers, a 30 hours course, which was well appreciated. Recently, few days back, on 25th of February, there was a science fair where these young children learned about science in a playway method, you can see that they are enjoying, each of them trying to see what smells and what tastes and how rough is the surface. So science education actually starts right from daycare to school students to college students and beyond. And for the school students, we have a small laboratory. Our volunteers show some experiments here. We have a science nurture initiative wherein students from neighboring school come in and they are helped with their syllabus by our volunteers who teach them whatever they have learned in school and help them to understand so that they walk out confidently. We expose them to different lectures. There's a science club. We run science exhibitions, which I will tell you about. And today you are sitting in on a popular science lecture series, which is meant for college students, supported by the Board of Research in Nuclear Sciences. We also run lecture workshops and refresher courses for the faculty, supported through the science academies. We have a learning garden, and wherein we have about 500 different types of plants. We take visitors come and see them. We take them out to exhibitions so that their society is made aware of the different types and characteristics of these plants, and it's interesting learning. And we have moved on with many more activities, which include skill development programs. We have green initiative on campus, which is solar system for electricity and solar water heaters for the whole campus. And during the pandemics, we continue to have some amount of online teaching. The projects in collaboration with Gyan Prasar include Each One Teach One, wherein a college student mentors or helps a little young student from a school to understand the syllabus that they have and if they've not understood it. This is teaching the college student to take on the responsibility, and the youngsters from the school enjoy learning with a little somebody a little older than them. We monitor all of this through our senior volunteers from the IFSA members. We've run this through different colleges up till now, and we also have a science club, which is with the Vigyan Prasar. It's referred to as the WIPNet, which has monthly experiments shown or lecture series, which are conducted here for the youngsters. These are from different schools. And we also have garden based workshops, which we have conducted under the collaboration with Vigyan Prasar. IFSA also runs internship programs for college students, and we've done them right around 5 of them, which have been conducted very successfully. For this, our volunteers from IFSA members give projects. Then there are expert lectures online, and then at each point the projects are evaluated by experts both nationally and internationally. And in one or two cases we've even come out with nice booklets of their outcome. So it's been a very great learning experience, not only for the students but the mentors also. We just celebrated Science Day at IFSA. It was on a topic called as biomimicry, where young children from schools from standard 6 to 12 could participate, and they could either do a PowerPoint presentation or an essay. And then these were then evaluated by well acknowledged scientists who spend their time to judge these presentations. And we also had Miss Sonam Ambe as our chief guest. And we learned from the youngsters how nature has so much to provide, and what we know as Velcro today actually has its origins within the nature stories. BRNS supported lectures. You see, we this has been conducted since the last 11 years. We are now in a 3-year program, which is 21 to 24. We had conducted lectures only in colleges like the one we are doing right now here. And during the past season, we reached out to nearly 6,600 individuals. 60 lectures varying from physics, chemistry, biology, environment, and engineering, all resource people from acknowledged academic institutes and very acknowledged speakers themselves. Today is our 30th lecture, which is being given by Dr. Susan Nippen, who, when you will see her introduction, you'll realize why we requested her to speak to you all and speak to you all on scientific writing. This year, we have also moved on to schools. where we conduct the lectures of interest on different topics. Here, one you see is on skill mathematics, and now we on site. Here, you can see the young children listening to something about blood and hematopoiesis. We also move out to society, where we have lectures of interest, say on marine archaeology or the ecosystem, and many others to follow. And we're going to have some lecture on James Watt telescope. So do stay connected to our website. You will get all the information. What I've not told you about is in our newsletter. You can download it, and you will also hear about what we do in the other 11 branches, which I have not projected, but they are very similar activities. So do stay connected with us. And before I actually end, our activities have been recognized by the UNESCO. In the last week or two, we had a visit from the UNESCO who have interviewed our founder, member, Dr. Sudha Padde, and our chairperson board of trustees, Dr. Sunita Mahajan, to learn about our activities. And they were on our site at Vashi, who have looked at all the activities and were very happy to be there with us. So we have been identified by the UNESCO also, and we are privileged today to have Dr. Susan with us here, who now is one of the recognized scientists in the um, Principal Scientific Advisors Board of recognized members. So you see that we try with all these efforts to reach out to society, right from daycare to senior citizens, and our efforts to see about knowledge sharing, bringing about a change, leading to value creation, and promoting innovation with emphasis on science based methods. And we request all those who become graduates who finish their third year, please do consider joining us as a member and join hands with us to join us with our activities. If you want, take a picture of this. You can connect with us and we can give you more information how you can participate in our programs. Thank you for being with us. And I'd like to thank the college authorities and Dr. Anupama in particular and Dr. Mr. Pushkar who have made this possible today. Thank you.
Thank you, ma'am. Now I would like to welcome our speaker for today's webinar, Professor Dr. Susan Nippen. Dr. Susan Nippen is a past president and member of Board of Trustees of Indian Women Scientists Association, a 15th batch BARC training school scientist in biology and radiobiology. She retired in the year 2011 as head Plant Biotechnology and Secondary Product Section, Bhabha Atomic Research Center, Mumbai. She did her special BSc Botany from Maharajas College, Ernakulam, MSc in Genetics and Plant Breeding from University of Kerala, and PhD from Mysore University. She is considered one of the pioneers in transgenic plant research, and her interest varies from plant biotechnology, production of secondary metabolites from in vitro cultures, and phytoremediation. She has published 140 research papers, contributed several book chapters, and presented papers in several national and international symposia. Her latest paper is on phyto extraction of uranium using vetiver plants. During an Indo-FRG bilateral program in 1986 at Free University Berlin, she developed the first transgenic grain legume from protoplast using three different techniques. Later, she developed transgenic plants which could withstand fungal and insect attack and take heavy metals and degrade organic pollutants. She was also involved in the Cotton Mini Mission project of Government of India and was a collaborator for several BRNS funded programs all over India. Scientists from Russia, Cuba, Syria have come to her lab to get trained with latest techniques. She had been in the editorial board of international journals In Vitro Plant and BMC Biotechnology. She received Elsevier Award in 2010 for the Best Quoted Paper of the Year. She is also a recipient of UNESCO and British Council Fellowship, Stanford University. Stanford University has recently included Dr. Susan Nippen in the world 2% top ranking scientist in the category plant biology and botany. She is one in the 75 women in STEAM in the list of the principal scientific advisor. It's a great honor to have you here today, ma'am. Before we begin with the webinar, a few instructions to the participants. I request all the participants to be kindly patient. If any technical problem arises during the lecture, we will solve all the technical glitches. Kindly mute your audio and video for smooth conductance of the session, and you can post your questions in the chat box at the end of the session. Ma'am, I request you to please take over the charge of the session. Thank you.
Good morning, everybody. Just a moment. Can you see my slide?
Yes, ma'am.
Is it in full screen? No,
No, it's not in the full screen.
just a moment. Now,
Yes.
Yes. Yeah.
yes. Thank you, Surekha, Anupama, Pushkar, and all the others from Patkar College and also IUSA members. Good morning to all of you. I am extremely happy to speak about scientific writing to students of Patkar College. In fact, if I had taken this class in person, I would have brought some reprints along with me, and we would have had a very good discussion, and you would have understood in a better way. Anyway, I'll do my best in this webinar so that you understand. So, when I spoke to, I'll come to that. An academic paper can be a research paper. It can be a case report or a review article, an update on the latest information in a particular field, or an opinion article, or it can be a project proposal or a project report or MSc thesis or PhD thesis. It can be anything. But today, I am asked to concentrate on a research paper by Anupama. So I am concentrating only on research paper, not on other aspects. So you should have a novel idea before you start the research. You may be given a problem by your guide or in the group you are put in. First thing for you to do is to get the literature in that particular field. Suppose you are working on COVID virus or some nanoparticle, gold nanoparticles or something like that, you should go to the library or you should take the electronic database and search, put keywords and search. That is a literature survey. So you will come to know how much of work is done in the particular field. Then you have to think, you have to discuss with your colleagues, with your guide, and then plan your experiments. And you have to do the experiments properly. Suppose you want to find out a new and a quick method to detect COVID-19, since the RT-PCR takes maybe 3-4 hours to detect the virus. So you have to find out new method. You are going to find out a new method. So you make a instrument or whatever you want to design, and you have to conduct the experiment, and then you have to take samples. You have to go to different hospitals, some in Mumbai, in Delhi, and Calcutta, and Chennai, and Bangalore, and maybe take 100 patients, do RT-PCR, and compare with your method. And is your method quick? Is your method reliable? 95% or 99%, how it is comparable to RT-PCR currently which is used, you test like that. And you collect data from all the places. Yeah, and you have to analyze the data collected from Mumbai, from Delhi, from Bangalore, and all that, and analyze them, and make into tables, graph, etc. And you have to project what the results you have got, and finally you have to write a research paper with all the work that you have done and with all the results that you have got. So why you have to write a scientific article? As a scientist, some of your students at the moment, you may take up a scientific career, you may be working in a particular field, and on viruses, you may be working on nanoparticles or in a plant biotechnology, or transgenic plants or CRISPR-Cas9. So as a scientist, whatever experiments you are doing and whatever results you are getting, you are supposed to share your research work with the international audience. Your audience is not just from India, but they can be from United States, they can be from Germany, Mexico, Africa, Russia, anywhere. So whatever work you are doing should be a long lasting reference to other scientists, because many people are working in the same field to find out like new things. So whatever work you are going to publish in your article will be archived. Maybe even after 50 years, you can take out the information and use it in their work. And where you are going to publish your research paper depends on the impact factor of the journal. I will come to that in the end. What is the impact factor of the journal? And if you publish in a journal which is having high impact factor, that will show that you have achieved quite a lot, reflects your achievements and your credibility. So, what is effective scientific writing? You are sharing whatever experiments you have done, whatever result you have got with not just one scientist, but with the international community. You are sharing your research. You are not hiding anything. There is no secrecy in the research. You are presenting everything for the researchers, so that others can read your work. You have seen buildings being made. There is a strong foundation, and you put a brick one on the top of the other. So the earlier scientists have already made probably the foundation, and they have placed a few bricks one on the top of the other. And you are going to add one more brick or two more bricks on the top of already built building, and you are depending on the work of previous authors. So the previous authors, what work they have done is very important. And when you are presenting, it is not like like in Times of India, you write an article on Russia Ukraine fight or something. It is nothing like that. You don't use any flowery language. It should be accurate, and it should be concise and precise. It should not be secretive. You have to write out everything. You don't want somebody to repeat, and you don't give all the methods and materials. It should not be cryptic. It should not be secretive, and it should not be self-centered. You should not give so much importance to you, what you have done. I have done like that. I went and collected the data. I went to Chennai hospital. Nothing of that type. The data was collected from Chennai or whatever it is, and you are addressing to two types of audience. One is the editor. You are sending the journal article to an editor, and it will go to the referees, who will decide whether your paper is worth publishing or not. Secondly, it will go to the journal readers. So it's very important that you should convince the audience that your research that you have presented is correct, it is valid, and especially for scientists who are working in the same field. Suppose I am working in the field of antiviral compounds from plants. All the time I'll be looking for literature, whether any new thing is coming up or not. So you should convince the audience who are in the same field. So why you have to publish in international journals? You can publish in Indian journals also. That is very good. But the impact factor of most of the Indian journals at the moment is very low. And maybe you can try to improve the impact factor of Indian journals. So when you publish in a good journal, you will leave a record of research. And when you are putting a brick Another person can build on the top of that, and the building will grow up and up. So the research of one person will be added on the top of others, and another person can add, and it will go on. And when you publish a paper, what happens? You will get recognition for the new ideas, for the new results, and you will attract interest from others in the particular field of research. And when you publish, you get feedback from different people. So many people will ring you up or contact you over a, like a WhatsApp, or like a email or something like that, and they will ask you, how did you get that? They will discuss with you, and you have to legitimize your research. And to legitimize your research, if you are doing an independent experiment in Patkar College, somebody in Ruya College should be able to repeat it. Somebody in Xavier should be able to repeat. Somebody in USA should be able to repeat. So you are to legitimize your research, and you should have independent verification. That means other people working from other labs should be able to repeat your experiments. So why difficult? It is difficult to publish because writing, as you know, is a skill. It is not like writing in a magazine or a story or something like that, because scientific writing itself is a skill, and you have to read many articles. Initially, you can go to some of the important journals and take out important articles which interest you, and go on reading it, and then improve your skill of writing. And many times you don't get positive results. If you get positive results, as you know, it is easy to publish than negative results. And each scientific journal has specific requirements. What is needed for Nature Biotechnology will be very unique, how they are presented. So go to each journal where you want to publish and see how it is published, how the papers are. You look at the different sessions, and you are participating in an international community. You are joining an international conversation, what other people are conversing and what work is done up to now. And you should get access to journals where in the people in the field are publishing. You can have email alerts. If you subscribe to some journals, they will send you the latest published information on the latest issues, and you will get the email alerts. And you have to have good the skills on internet and electronic database searches like Scopus, and you can search, put the keywords and search what's the latest information. And attending international conferences will also help, because many times they don't publish. When this CRISPR-Cas9 was developed, the two ladies who got a Nobel Prize, they met at a conference and discussed. So a scientific manuscript should have the following sections. What are the following sections? It should have a good title. First of all, the title should be very good and attractive, and it should have a very short and crisp abstract. Then keywords. Keywords will show the main points of your work. I will come to each and every session in the later part of my talk. Then the next one is introduction. Why you have done it, what you are going to achieve. Then material methods. What type of material you have used, what type of experiments you have done in material methods. Then results. What are your results? What tables you are presenting, and your text you have to present in the results. And so in some journals, results and discussion may be together. Usually it is separate, like discussion. In discussion, what you do, you take out your results, and you have to compare with the earlier work, and what's the new theory that you are getting, what's the new result you are getting. You have to discuss in that. And some journals will have a short conclusion, and then references and literature cited. Okay, after that there may be some acknowledgement also. Now, how you are going to start writing a manuscript. What is a manuscript? It's a journal article or a research article. So there are different types of people you have to address. One is the people working in the same field. They know all about the field. they want to know more about the field, what is the latest in that. So you are addressing those people who are working in the same field. And some people out of interest, like suppose it's a medical biotechnology journal or something like that, they will have interest, like medical people will have interest, pharmaceutical people will have interest. So people from relative fields also may read your work. So you are going to start writing a manuscript, and reviewers, they know this topic very well, and they will do the standard screening. And English language you have to improve, because ours, our native language, we many of us learn in native language. And even if you learn in English language, the writing of a scientific in a scientific journal is very different from writing in a normal article. That first thing that is needed is a very good title. So what is a research paper title? Is a very important communication with editor, and then from the editor to the referees and to the readers. When they see the title, they should feel like reading. It should be a good title. You should identify the field of research, whether you're working on nanotechnology or whether you're working on plant biotechnology or CRISPR-Cas9. You should tell the title should indicate the story or the results that you're going to tell later. And when you read the title, you should raise questions about the research in the mind of audience who is just looking at the title. Then you want to read more about it, and it will attract the target audience. Some people are always looking for a particular field, and when they see the title, they want to read more and more. So more revealing the title, more easily your potential readers can judge. If the title is very good, your readers will really want to read your work. But the title should be concise and informative. The number of words are limited, like 8 to 12. It depends on the journal. You can go to the journal and see how the title is for different journals, and you should use some particular keywords in the title which will capture the attention of readers, and it will be used for indexing in Scopus and other things. Now, I am just giving a few title. I just made this title. I may not be correct. Example, I'm giving title: an effective antiviral protein against SARS-CoV-9 from Ophioryza rugosa. So, what does it? What comes into your mind when you read this title? It shows that there is an antiviral protein against the most important pandemic cause, that is SARS-CoV-9, and it is a plant origin. So Ophioryza rugosa is a plant, and from that you can isolate a protein against this virus. When you're reading the title, you understand, what is going to be presented in the paper. Another example, Molnupiravir, an effective oral treatment for acute COVID-19 pandemic. When you read this, you know that those doctors and clinicians will come to know that this particular medicine can be used for treatment of acute COVID pandemic. And remdesivir, or that medicine, you all would have heard about it during the last 2 years, to prevent COVID-19 progression in outpatients. Then another title, tissue engineering of uterus supports live birth of rabbits. What does it show? That you can do 3D printing and tissue engineering. You can make an artificial uterus, and it will help in birth of rabbits. Maybe it can help in the birth of human beings also, probably. Then another title I have just showing here is a male biased sex distorted gene drive for control of human malarial mosquitoes. Man is always fighting against mosquitoes. Is there using CRISPR-Cas9 and a male distorted gene drive? Can you control human more malarial mosquitoes? So you want to read more about it. So how to improve the title? You may write a title and write 3-4 times in 3-4 different ways, and then you see which one is best. I have just given one example in 2 different ways. Effects of added calcium chelators on salinity tolerance in tomato. The same can be written in another way. Addition of calcium chelators improves salinity tolerance in tomato. Or you can even we write in another way, uh, uh, improving salinity tolerance in tomato by addition of calcium chelates. Which one is better? You 3-4 titles you write and look, discuss among yourself and select the best, most impressive title. Now we come to the second part, that is a abstract. 1 minute. Sorry, I think there was a mistake. Now, title restructuring. When you make a title, keywords should be put in the front of the title. Then you should have a colon, the 2 dots, or a dash. The main title you can divide into 2 parts. The most important part on the first part, and the secondary part later. For example, just have a look here. Disturbance, invasion, and reinvasion. This is the main part. And here you are putting a colon, management of ecosystems. Or you can make phytoremediation of cadmium from soil. Then you can put a hyphen and you can write improvement using chelates. So you can restructure the title in such a way that it is made into 2 parts, and connecting them there's a colon or a dash. Now will come to abstract. I think it has gone to the other side. Busy people read only abstract title and keywords. They don't read the whole thing. Like nowadays, everybody is so busy with the WhatsApp and so many things that you don't have time to read. So when you see the abstract, you usually spend only 1 minute. In 1 minute, you should understand what is going to happen in the paper. So busy people read only abstract and spend only just 1 minute because they're really busy. And for some journals, like in developing countries, like many universities and all, you cannot get the full paper because only abstract is available. Then abstracting services are available, and you can use that title, keywords, etc. And you can go to different databases like Scopus and all that and get information. In our time, when I started research, we had to go to the library and sit for hours and and scan through biological indexes for several years. Now things are very easy. So the maximum number of words in an abstract should be only 250 to 300 words. It can vary from journal to journal. What should the abstract have? One line on what is the background information. Why are you doing this project? Why are you writing this research paper? And what is the principal activity of the study? What is the scope of this study? And something about the methods used. If you want, you can write about the method used. It's a new method. And what is the most important results of the study? And then what is your conclusion? What is your recommendation based on your results? So abstract should have these particular points, but it should be only 4 or 5 sentences, and the whole message is conveyed to the audience. So I have just written an abstract for you to read. Severe acute respiratory syndrome, coronavirus 2, in brackets SARS-CoV-2, the virus causing coronavirus disease in 2019, COVID-19, was reported first time from Wuhan, China in December 2019 and became a major health hazard throughout the world. Early diagnosis is crucial for patient management, outbreak of control. So most tests currently used for detection of SARS-CoV-2 rely on RNA amplification using real-time PCR, RT-PCR, and require few hours. Hence, a highly sensitive, why you are taken up this problem now? Hence, a highly sensitive and a quick method is essential for rapid detection of viral antigens. So since it is taking a long time, you want to develop a method which is very quick. So in the present study, we used a new method on colloidal gold nanoparticles linked to monoclonal antibodies fixed to a nitrocellulose membrane which detects COVID-19 quickly, and the results are presented in the particular paper. So People will feel like reading. The clinicians, doctors, those who are working on COVID-19, they want to read immediately because a new method for detection, you don't have to wait for 4 to 5 hours. So this will be abstract, will be followed by keywords. What are keywords? Each paper should have 4 to 6 keywords. These keywords will be used for indexing by these journals, by databases, should give what the research paper is about. For example, I gave this title before, a male biased sex distorted gene drive for control of mosquitoes. What should be the keywords here? Gene drive. I don't know whether you have studied gene drive. Anyway, CRISPR-Cas9, all of you know that this is also there, part of it. CRISPR-Cas9 gene editing. Then it is male biased, mosquitoes controlled. Mosquitoes will be killed, but only males will be killed. So there will be no mating. Then Anopheles gambiae is one which was used in the study, and Plasmodium falciparum is the agent which inhabit this Anopheles. So the keywords, when you read, you know what it is about. Now we come to abbreviations. You may use many abbreviations in the text. Whenever you are using an abbreviation in the text, when you are starting writing the paper, first time when you are using, you should use the full word and then give in the bracket. And many journals have a footnote where the abbreviations are given. Like you are used SARS-CoV-2, and some people may not know what is SARS-CoV-2. So you have to write in the abbreviation severe acute respiratory syndrome coronavirus 2. RFLP. What is RFLP? Some people may not know. So restriction fragment length polymorphism. KN is kinetic. 2,4-D is 2,4-dichlorophenolic acid. CRISPR is clustered regularly interspaced short palindromic repeats, etc., etc. This will be given as footnotes or sometimes just after the abstract or keywords. Now you come to introduction. Introduction is a motivation. Actually, when you read the introduction, motivation of work, and the readers should feel like reading. When you read introduction, you have gone through the title, you have gone through the abstract keywords, and the person who is going to read is wanting to know more about this. So he comes to the introduction. So it motivates the readers, and the reader should feel like that they want to continue to read. So effective introduction is necessary. The editors will read, it will go to the referees, it will go to the audience, and they all are going to read. So what will the introduction address? The introduction should show that your contribution is new. You are not just repeating somebody else's work. Is the contribution significant? Is it suitable for publication in the journal? The editor will look whether the paper that you send to that particular journal is suitable for publication in that particular journal. So if you are working on nanotechnology, you cannot send it to a plant biotechnology journal. The editor will screen and then immediately he will send it back. If the paper is good, he will send to the referee. So introduction is important. So 5 stages of introduction. What type of field of research you are going to do? A statement is needed in the introduction to provide readers with the context for the problem to be investigated. Why are you going to do the research? Why are you going to write this paper? Like in Wuhan, there was a release of this coronavirus 19. That's why you are going to do this problem. You are dealing with the problem. Then there should be more specific statement about the problems already studied. Some people who have already studied, they have developed methods for detection. 3-4 methods will be there that you have to quote in your introduction and give the reference in the bracket, like Smith et al. 1920. Like that, you will give the reference in the introduction. Somebody has done some work previously, you have to quote in the introduction. So you are laying a foundation of information which is known already, but the reader may not know. So you have to give the quotation for references. Then you make a statement. Why this work needs more investigation? Because there's a gap for the presentation, for the present study. There's a gap for the information available, and you want to fill the gap. You want to fill with the new information. And why are you doing this? What is your objective? What is your objective of the present research? And then outline the major activities and finding 1 or 2 sentences you can write on what are the new results you have got, and then you can maybe you can what is the further research we can suggest if you want to say optional statement. So stage one, what type of tense you can use in the introduction, whether you can use past tense, present tense, or future tense. And this statement is accepted by members. All the power things which are already reported that from Wuhan, some Wu et al. Have already reported. So it should be in reported. It is in past tense only, because you are quoting somebody's work in the beginning. So it is in the past tense. Okay. So in the stages 2 and 3, old information the reader already knows about it. Okay, so that is placed in the beginning. And new information will come in the end. So select literature. You go to the literature. It is very important that you go to different papers, read it completely, understand it, select literature to justify why you are taken up this study, and you should construct a niche for your own work. Sentences, each sentence, if you are taking from somebody's work, it should be supported by references, whether it's a research paper or an article, review article, book or website or something. You should always give the reference. Otherwise, there is a big problem. I will come to that. Then after introduction and why you are doing the work and all that, what earlier people have done, then what is the main result, why you are doing it, you have already written in the introduction. Now what are the materials you have used, what are the methods you have followed. Okay, now what is the purpose of this material methods section? Because I told you that you are doing the work in your college, Patkar College. It should be repeatable in Miranda College in Delhi, in a University of Nottingham, in IUP in Pennsylvania, USA. So it should be repeatable. So the method section should be perfect. If somebody wants to repeat, don't hide any fact. So many scientists, after reporting publishing some papers, you find it very difficult to repeat after reading the material methods, because you have become secretive. You are not given the complete information. And if some material methods in material, somebody has published like protein estimation, Laurie's method. If you are used, you can give the reference in the bracket, give credibility for the information. And what are the referees looking? When the referees are looking, reading your paper to decide whether to select or not, they will think, do the methods and treatments really conform to acceptable scientific standards? There are acceptable scientific methods to look for, do things. Are you doing the correct method? The referees will look. Can the methods, material and methods, experimental procedures, repeatable? So methods published earlier, you don't have to perhaps give in detail. You can just give reference in brackets. Sometimes it is in a name and followed by year, or sometimes number. Okay, so you have to give the information which is available in the material methods. So normally material methods organization comes before results. So after introduction comes material methods and then results. So material methods, this section comes before results. So you can even have subheadings, like in material methods, you have collected material, you have collected the plant from Mahabaleshwar and all from Himalayas, okay, and brought it to the lab and sterilized it and do it whatever it is, extracted the chemical, that you can write. And then you can have subheadings in the material methods, and how did you extract, what procedure you used for extraction, and what how did you detect the chemical by HPLC method, okay, and which instrument you have used, and if you have done RT-PCR, which instrument you have used and what method you have used, and all the details you have to give as subheadings. Also you can give at the end of introduction, and the same headings can go into the results also. In material methods, you can write a few, 1 or 2 sentences, and each you can have subheadings like a detection of the phytochemical, then isolation, all those things you can give. What type of verb we can use? Many use passive voice, some use active voice. For example, the dog bit the man. I'm just giving an example. It's an active voice. The man was bitten by the dog is a passive voice. So passive voice is normally preferred because you don't want to say about the agent. So you don't want to say the agent, and so you use passive voice. So don't use the word we. Okay, use the simulation modeling was done, etc., etc. So don't use it was done. Don't use we did it or I did it. So active and passive voice. One example: the researchers collected data from all sites weekly. It is an active voice. It is not important who collected the data. When you are doing, it is known that somebody is collecting the data. So just change into the particular voice, passive voice. Data was collected from all sites, from Mahabaleshwar or from wherever it is, that you have to write. Don't have to write that my assistant went and collected or the researcher went and collected. You don't have to write. Data was collected from all the sites, or the data was collected from Mahabaleshwar and Chennai and like Nagpur and all that. So second example I given is be calculated least significant differences, that is LST, to compare the means. You don't have to use we the word, just change it into a passive voice. That is a passive voice is least significant difference, that's LST, was calculated to compare the means. So omit words like I, we, etc. Now you come to the results. So you have taken the observation from different hospitals and you have compared your new method, you have compared with your RT-PCR, how effective it is, how repeatable it is, all those data you have collected. So much data from one hospital, Nanavati Hospital in Mumbai, then another hospital in Delhi, then Chennai, Bangalore, and other cities you already collected. Now, how to start your results. Results is like writing a story. You write, like I think as children you will be given small words and you will be asked to write a story like that. You are going to make a story out of the results. Writing as a story is a key driver of an article, and the article should have one or two take home messages. What is a take home message? And then first you have to make the table. You collect the data, you make a table and figures first, then only you write the text. Based on the figures and the tables, you write the text. Sort out the tables. You don't have to put all the data and the figures in the table, and then connect all the things and write the results. And you are maybe having many co-authors. Your guide will be there, and you can discuss the story of your paper with your guide. You can sit around a table and discuss over tea, and what data to be included. Do you have to present all the data that you collected from all the hospitals, or only few data that you have to discuss? What are the important points from the story you have to discuss, and what is a take home message? So while making the results, what do my results say? You all of you sit together and write a brief summary. What is the result? What is your main finding? Okay. What do these results mean in this context? What is the conclusion that you will draw? That it's your technology, your technique of detecting COVID within 10 minutes is as good as RT-PCR. Is this the conclusion? And now the audience want to know more about the results. So what contribution with the results make to ongoing results in the field? Suppose somebody is working in the field, what is your contribution? What the results or what your results are going to contribute new to the field? So if the audience are not able to read your paper, what are they going to miss it? So the results is turning data into knowledge, data into text. So you should know good statistics, and you should analyze the data and present your evidence to support or reject the hypothesis. Your method is good or your method is bad, method is worse than RT-PCR or not. That you have to do the tests of significance, analysis of variance. Whatever test you are doing, you have to do statistical test, and you are going to present in tables and figures. And in the tables and figures, you give some numbers: table 1, table 2, table 3, and figures also: figure 1, figure 2, figure 3. And these will be numbered sequentially, And when you are writing in words in the results, you should give the number in the bracket, the table 1. You write one sentence and write in the bracket table 1, table 2, or figure 1, figure 2. So in many major journals nowadays, what happens is so many new papers are coming, and they find it very difficult, to include all the things in the journal, all the results or whether material and methods. They cannot include everything. So what do? They have additional data as a supplementary information, and there will be a link given there. No, like you don't want to present all your tables. You give the most important table and most important figure, and there may be another 5 6 figures which are according to you important, but not so important for the journal, because they don't have time to read. People don't have space. Journals don't have space, and people don't have time. So those who are working in the field will be interested. So all these tables, extra information can be given as a supplementary information or supplementary data, and when you press the link, you will get the information in the journal site. So data presentation vary from a journal to journal. So you have to use your data to support what you are writing. So tables and figures should really support your results. Tables and figures should stand alone. Suppose you're reading only table, and when you see the table or when you see the figure, you should be able to understand what it is without going to the text in the results. You should be able to understand the data presented in the table, because there will be a legend, and when you read the title or the legend of the table, legend of the figure, you will understand the whole thing. There is absolutely no need to go to the text. Without going to the results text, you will be able to understand. So how the day you go to the particular journal where you want to publish it, and then you present your data accordingly. Refer to the latest article. Each journal will have, like how to, they will have instructions to the authors. Go to the instruction to the authors, and what is the choice of tables and figures? Is table better or is figure better in this particular journal? What have they given? Then you decide your mode of presentation. Okay, figures and tables, choice of figures, tables are yours. Figures or tables will depend on the point you want to convey. You are the person who want to convey something to the readers. So readers have to depend on what you are presenting. So whether you are presenting in a table mode, whether you are presenting as a figure, depends on you. So tables will be useful because you want to know little more about the data, like 10.12. Then detail, that's decimal also you want to know, then table is useful. And calculations, explaining calculations or showing components of calculated data, tables will be useful. However, the actual data is given in the table, so it will give a real comparison between different elements in different direction when you use a table. So tables have some advantages, but figures will be useful because you just want to see a trend, the growth curve, curve it is showing how it is going up after 3 4 days. So it will show an overall trend or picture. It's a comprehension of theory through shape rather than the actual numbers. In table, actual number is given 10.12, whereas in the figure it will be just a shape. How the shape is given more importance in figures. So allowing this will allow simple comparison between only a few elements. So choice between figures and tables is for you to decide. When table will be useful when you want to know the exact number, and figure will be useful when you want to show it as a shape only. And individual data values are important when you are concentrating on individual data values. Then table is important. Whereas when you want to see the overall pattern, figure is important. Okay, you want more precision and accuracy, table will give more precision and accuracy, and in figure it is just an overall picture, so it will be not so important. So I am not going to the details. So So designing tables, you have to decide which row, how many columns, how many rows, which are there. You might have collected so much of data, but you need not present all the data, only the important data. You would have collected data on the 0 day, then first, second, third, fourth, fifth, 10th, up to maybe 1 month. So you don't have to collect, give in the graph all the details of the second day, third day, fourth day. You can say 5, 10, 15, 20, like that you can make. So the choice of for data inclusion, arrangement of data within the table, it should be clear and informative, and column headings should be given on the top, and table title should be there, and it should not be very there should be no clutter in the table. Keep the tables free of clutter, and put a footnote at the bottom of the table, and the table should have a clear title on the top. And unnecessary data is there, you remove the data and throw it off. Or if you want, you think it is very important, give us a supplementary information. Only if those who are interested in the real work, those who are working in the field, will be interested in supplementary information. So in the table design, you can see are all data needed, and you is a title description informative, are the all the numbers like 10.53, is it rounded up 5 up to 10.5 to show precision, or you want to give 10.53, or does the table have any other weakness? So you can figures and legend titles are very important. I said all the figures and tables should have titles, and it should stand on its own without going to the text. One should, when you see one table or one figure, you should be able to understand what is the data presented there. A figure legend should have 5 parts. A title which will show what the figure is about. The title of the figure will show what the figure is about and what are the results or models. Then additional explanation of different components of figures, methods used can be given in the title. Description of units, statistical like standard error you are used, or analysis of variance and LST, whatever statistical notation, statistical analysis you are used, you can write. The results were analyzed by statistical analysis. You can do explanation of notations or symbols if you are used in the table and figures. Here, this is a figure of a bacteria. So this is given figure 1, and then you give the text, the particular bacteria under the microscope or whatever, and this will be referred in the text as figure 1. Then you want to put a table. Suppose you are measuring plant growth in soils with the different pH values at 6, 6.2, 6.4, up to 7. What is the height of the plant? Average plant growth. You have to write there. Then you have to include the statistical analysis like standard error or something also here. And when it's a graph, you have isolated a particular compound. The red one is negative control, and the other two are 2 drugs isolated from plants, positive control, and within maybe within 20 to within 10 to 20 days there is a complete death or whatever it is, like so the effect of the drug is shown here. Graph is very useful. You can have different types of bar chart, and you can stick one on the top of the other. It is for you to select what type of a figure you want to present. You can have a pie chart. So statistical analysis is very, very important. Without statistical analysis, your paper may not be accepted at all. So the science of collecting, exploring, and presenting large amount of data to define the pattern, underlying patterns and trends is very essential. So I am sure that all of you would have studied, done some statistics in your college. So you know what is standard deviation, standard error, statistical significance, correlation coefficient, analysis of variance, student t-test, chi-square test, etc. So some information has to be there. The data that you have collected should be analyzed statistically, and that has to be presented along with the table or figures. Now, writing about results, do not repeat the same words again and again in the results and figures. Write only the most important findings. And in some journals, I said before that results and discussion may come together. Results are things that you have done, whereas in discussion, you are comparing your work with the work of previous authors. So separate or together. If it is separate, is better. So check the instructions for the journal for the contributors and decide whether results and discussion are two different chapters or they are together. And if you are writing only in the results, only results, then don't compare your work with somebody else's work. Don't compare your work with somebody else's work. You present only your work in the results. Whereas if the journal wants results and discussion together, you present your result and compare with that of previous workers. So, what are the functions of result sentences? Result sentences will highlight whatever findings, what are the important findings in your research, and your figure and tables are given, and then comments also will be given. These are the functions of result sentences. Then, what type of verbs can be used in the results? Usually, past tense is preferred to be used in result session. For example, was used, were raised, identified, did not cross react. Past tense can be passive or active. Okay. Sometimes when the situation is very real, you can use is. Okay. Otherwise, for example, uranium is a common radionuclide. It is everybody knows. Even yesterday also it was a common radionuclide. Even today it is a common radionuclide. So you can use the present tense. It depends. Well, usually past tense is used because you already conducted the experiment. That experiments were conducted and data was collected from different places. Seedlings were raised and phytochemicals were identified. It did not cross react like that. In the past tense is used in the result session. But if it's a very common knowledge, present tense is used. Like uranium is a common radionuclide. Even every day it is a common one. Now we come to the discussion session. What happens in the discussion session? Does the journal, I told you before that some journals have a combined results and discussion. Would this suit your story? Does the discussion section include subheadings? Does the discussions results also can have subheadings? Okay, discussion will connect the issues raised in the introduction with that of what you have found. So do not repeat information. What you have written in the results or in the introduction, don't repeat it in the discussion, but you can include and conclude. So discussion should does it include subheadings? Is there some journals have a conclusion session? So review whether they support original hypothesis, you have found out something new, and how they contribute to the main activity of the study, whether you agree with the findings of earlier workers. Explanation, all the references also are given, and in the end of discussion, what is the importance of your finding, what's the implication of the study, and what is the recommendation for future, what are the practical application. You have developed a new novel technology for detection of COVID-19 quickly and with 95% efficiency. So what is new in this? So you should discuss it in the light of earlier information available, and you should give the references in the bracket according to the convention of the journal you have selected. Now, what are the possible verbs to strengthen? You can use better verbs. Suggest, instead of suggest, you can use demonstrate. The experiments demonstrated. Maybe stabilized. Don't use maybe. You can say is stabilized. Strong words. So, common problems with writing passive sentences: do not make too long a sentence. For example, wheat and barley samples collected from Punjab as well as ragi and wheat from Karnataka were used. Make it little shorter. Change to wheat and barley collected from Punjab and rice and ragi from Karnataka were used. Don't write it. Make it shorter. Or 4 cereals were used: wheat and barley from Punjab and rice and ragi from Karnataka. So this way you can shorten the sentences. Then use abbreviated sentences. The data were collected and they were analyzed using something. Okay, it's better to write data were collected and analyzed You say, okay, don't, and again, you don't have to say they were analyzed. Data were collected and corrections were calculated, changed to shorter ones. Data were collected and corrections calculated. Don't have to use were calculated. Okay, so like that you can make shorter sentences. And references, how do you give? There is a session for references, literature cited. Like for example, suppose there is only one author, you have to follow the format of the journal. Now I am just showing a general one. If there is only one author, maybe Caudle, GA is his surname and initials. Then 1974 is the year of publication. The biosynthesis of indole alkaloids, Lloydia, 37, followed by a colon, 219 to 98. In some cases, 219 to 298 it is given. Depends on the journal where you are going to publish. Della Porta SL and Hicks JB 1983. Two people are there. So between this thing, there's an and there. But the depends on the journal. Like you give in bracket the year, a plant, DNA, mini preparation, plant molecular biology, 1 followed by colon, 19, then 21. You write 21, not just 1. Okay, then suppose there are more than 3 authors, what do you do? Some of the journals you don't want to give so much of detail about authors. Wu, SF et al, because 4 or 5 authors are there. So in 2008, Camtooth is in 4 decades of surprising findings. Phytochemistry, journal number is 65, volume number is 65, pages. Sorry, here it should be a slight mistake is there. 273, it should be having 345. Okay, Seng MH. Suppose sometimes it is a volume in a book, a journal, some article in a book. Seng MH 230 to 2013. Camtooth is in chemistry. The book's name is In Plant Anti-Cancer Compounds. Who is the editor? Yamasaki Y or whoever it is. And what's the volume? And who is publishing it? Oxford Publications in UK, and which journal? What's the page number that you have to write. So the references have to be given correctly. And you have to give the acknowledgements. When you are acknowledging, support Department of Biotechnology has funded your project, your work. We can write the authors are grateful to DBT for funding, or we are grateful to DBT for funding. And somebody from IUP, Pennsylvania, USA has given you hairy root cultures. Then you thank that person, D.A. Lawrence, Dr. A. Lawrence for IUP, Pennsylvania, USA for supply of hairy root cultures. And Dr. S. Misra is a boy, like you are a head of the department. He has suggested something, so acknowledge him if he is not an author. Okay, now coming to the contribution of different authors. Recently, many people, many journals, you have to mention what is the contribution. Suppose there are 5 or 6 authors in the paper. What is the contribution of each author? For example, you write only the CRB conceived and designed the experiments. CRB is a short form of his name in the authors. He only conceived the idea and he only told them what to do and designed the experiments. Whereas DD is a full form for some name. GFSC performed the experiment. They are the ones who did the experiments. And WR did the electron microscopic study. And JPH analyzed, somebody else analyzes, did the statistical analysis. So you have to acknowledge not acknowledge, you have to mention what is the contribution of different authors in the published paper. Not all journals ask this, but there are some journals like Proceedings of National Academy of Sciences, etc., which ask these questions. Then I said you can have a supplementary information. Journals, as I said, have limited space, and then now thousands of articles are being sent to them. Some others can be given in the supplementary information. So many figures, many tables can be moved on to the supplementary information. The web links for supplementary information will be given along with the text or at the end of the text, which can be opened by those who are really interested. Now let me come to plagiarism. What is plagiarism? Is using data, ideas, or words that originated by another person without acknowledging the source. If you are taking out a sentence from somebody else's paper, you please give the quotation. Otherwise, it is plagiarism. If even a 3 or 4% plagiarism is there in your paper, the paper will be just rejected. And if you have already published the paper, it will be retracted. It will be very bad, not only for you, for your career, but also for the guide and also for your institute. Like some of the big institutes, the researchers, like students and all, they'll be just copying and pasting. You all are very good in copying and pasting, so don't, whenever you are copying and pasting or doing something, please give in brackets the quotation. If you don't give, it is considered as a jism or cheating. And there are mechanisms to check the plagiarism nowadays. Even in big institutes, they have it has to be checked for plagiarism before sending any paper. Paper will be rejected if plagiarism is detected nowadays. But there are some old papers where people have copied without giving the proper citations, and they need to be retracted. It's a very bad name for some of the prestigious institutes in India also. Incomplete citation, because a researcher must be writing the paper, and the guide, he may not read the whole thing, like he may not know whether he has copied from somewhere or not. So incomplete citation also will, is very bad. So avoid plagiarism. 1 or 2% maybe they will tolerate, but not more than that. Plagiarism, avoid plagiarism. Develop note-taking precautions. Okay. Now, if somebody has done the work, give the quotation. You should, as a scientist, you are supposed to give citations. Like Russell and Misra stated that uranium in blood samples. That means it is their work. Whatever work they have done, you give them credit for that. And because referees also know, they are working in the same field, so referees also know. So if you don't know, we don't write as your sentence, the referees also may know sometimes then software is to detect plagiarism. So avoid plagiarism. Now, how do you want to get your manuscript published? You have to select a target journal. You have to look into the journal and see whether they publish your kind of work, and check the impact factor. If you have got a higher impact factor, better for you. It will enhance your international credibility. Also, you should look for the time needed for publication. You look, in the top of the article, there will be the particular article was received on 1st January 2020 and published in 2021 October. So it has taken a long time. So does it take a long time for publication? When is a date received and when is a thing given? Then you look at the editorial board and discuss with your colleagues. Your colleagues will have experience of publishing much before you. They may have experience with each journal. So which journal we should select? You should have a discussion with your head of the section and also with your colleagues. And how to get most of the publishing? You want to publish quickly. If you delay by a few days also, it becomes obsolete. Publish in widely read journals. And see, first it go to the editor will look, whether you should go to the referees or not. Then he sends you 2 to 3 referees. And nowadays it's all electronic submission. In our olden days, we had to send it by post to the journal. It will take another 2 weeks to reach UK, and then it will take time to come back and all. Now it is all easy. It's all electronic submission. Editor will see whether it is suitable for the journal or not. He will filter it out. If your article is not good, he will immediately send it back. If it's good, if feels good, he will send you maybe he will give it to one associate editor, and associate editor will give to 2 to 3 reviewers. There's sort of blind refereeing. There are authors you don't know. As an author, you don't know who has seen who is your referee. Sometimes there's a blind refereeing. That means the editor will remove the name of the authors, of the paper, and the referees also don't know who has written the paper. Okay, and you also don't know. So double blind refereeing. And then the referees will send back to you that either it is rejected, and if you want to submit, what are the suggestions to improve? Do one more experiment, do one more concentration, and then you send back to the referees, resubmit. And your comments of the referees are very important. It will be either accept or regret and reject. So referees also have got some guidelines, for each paper they are assessing. The questions they have to answer is, does the title clearly indicate the title of the paper? Is the communication new? These referees have to answer yes or no. Is it suitable for publication in the journal? Is the organization accepted? Whatever you have written, is it the organization table, all those things, are they acceptable to the methods of treatment, like results and methods of material and methods? Is it okay? Do they conform to acceptable scientific standards? Or conclusions firmly based on data presented? Your conclusions, are they depending on this thing? So also the referees will have to answer, is the length of the paper satisfactory? Does it exceed 2 pages or 3 pages? Each journal has their own length. So are all illustrations required? Are all figures and tables necessary? Are all figures and legends and table titles adequate? Do title and abstract indicate content of the paper? Are references up to date, complete? Journal titles correctly abbreviated? Statistics have they done? Paper is excellent, good, bad, or whatever the referees have to do. And how do you select a journal? You want to select a peer reviewed journal, means it is gone to the referees, or just a open access journal like electronic journal. And many a time in colleges and all, they want to publish papers because maybe they are doing PhD and they should have two to three publications before you submit it. And they make the students pay, and even there are some insignificant journals, African journals and Romanian journals and all. They make the student pay there and publish it, and it has absolutely no value actually. So you have to get to publish in a good journal. It has to be done early and quickly, and choice of the journal, size of the audience, etc. You have to check. Most cited in the introduction. Okay, now let me go to the impact factor. What is impact factor? Is the average number of times an article published in the journal in the last 2 years have been, I'll come to that, discussed in a particular year. What is the time of publication? Now you would write a covering letter to the editor saying that you believe that your paper is within the scope of the journal, and state the title of the manuscript. You write that such and such a paper I am submitting with the names of the authors, and say that your research is original, you are not copied from somebody else, and what's the novelty of your paper, and express your presentation is satisfactory, and you look forward to acceptance and referees comments. And suppose it's coming back, be polite. Okay, and accept. If they suggested something, please do accept. Show and then you are resubmitting after doing one more experiment as suggested by the referees. Then show the editor that you have modified and revised the manuscript. If it is rejected, don't feel bad at all. Even Einstein had got some of his papers rejected. So if your paper is rejected, if it's really good, don't feel sad. You can consider another journal. So everyone gets rejection. So don't get disappointed. So improve your English skills. You have to read quite a lot. Improve your grammar, writing skills. You should have journal clubs, working groups, and you should go on reading new articles. And now, what is impact factor? It is a scientometric index of journals. Yearly, the mean, now we'll come to the details. Yearly mean number of citations of articles published in the last 2 years in a given journal. It shows, suppose a journal is having a high impact factor, like some journals like clinical, what is it, new journal of England journal of medicine, maybe 90 or 100 impact factor is having now, because everybody is working on COVID-19 and most good results are coming in that particular journal. If a journal is having high impact factor, it shows the journal is very important. So journal with high impact factors means more prestige for the author. Okay, and for the institute also, usually you get an impact factor of 2 or 3 or something. That is what normally happens, like or less than one also, but usually in a good institutes maybe 2 or 3 only usually. And then the impact factor is a citation in a particular year divided by publication in the year 1 year before that, plus publications in 2 years before that. For example, you want to know the impact factor of a particular journal, that's Nature Biotechnology or something like that, in 2017. That is citations in how it is calculated, impact factor of Nature Biotechnology in 2017. How many citations are there in 2017 divided by total number of publications in that particular journal in 2016, and total number of publications in 2015. Out of that, how many are cited in 2017. So, sorry. So Nature, so suppose the citations are 74,090. Previous 1 year before that, 880 publications were there, papers were there, and 2 years before that, 902. So impact factor is 42, for example. So initially, this impact factor was used by libraries to decide which journals to buy. Of late, it has become a measure of academic excellence. So, impact factor of a journal can vary from field to field. Suppose it's a clinical microbiology, you can have high impact factor. But suppose it is a very unknown field, you can have a low impact factor because very number of people may be quoting from that journal. Okay, so I'm just showing an example. Nature methods, nature biotechnology. This is not the current one, maybe 2014 or 13 or something. Nature, they have a very good impact factor. It can vary. PLOS medicine, BMC biotechnology, plant biotechnology, trends in biotechnology. Okay, then impact factor of microbiology journals. You children are from a microbiology and biotechnology background, so microbiology journals can have high impact factor. If 25 and all is very good. See, clinical microbiology, trends in microbiology, etc., etc., microbiome, etc., etc. Now, what is H index? H index is the author level metric which measures. When I was there, this was also not there. I don't know. Maybe it was there. I didn't know about it. Author level metric which measures the productivity of citation impact of publication. For example, George E. Hirsch initiated this thing. So it is called the Hirsch index. His surname is Hirsch. So it is called H index or H number. H index is defined as the largest H such that articles have H citation each. For after 20 years, if you have a H index of 20, it is very good. After 40, it is excellent. And 60, it is truly exceptional. Yesterday, like I also did not know what is my H index. No, I have retired 10 years back. So I just looked what is my H index. And I found it was 39.5 or 6 or something like that. So I was happy. Okay. So that is it. Thank you.
Thank you so much. Can you hear me?
Shweta,
Ma'am, this
who will
was.
be taking the questions?
Hello, can you hear me, ma'am?
Hello.
Thank you,
Shweta?
ma'am. Yes, am I audible?
Yes.
Thank you, ma'am, for enlightening us with your knowledge and experience. Participants surely got deep insights into the topic and also relieved some interesting facts. Now, with your permission, shall we proceed ahead with the question answer session?
Yes, yes.
Okay, ma'am.
Yes, ma'am. Okay, ma'am. Thank you. It was a very informative session. It will be helpful for the students who are doing their dissertation now and for other people who will be involved in future research works. So, I request participants to put their question in the chat box. I request all the participants to put their questions in the chat box.
You can write review articles also, like in the latest thing, like some COVID research or something, anything. CRISPR-Cas9 for cancer treatment, like that. You can collect the information done by other people. It's not your article. You can write a review article also.
Yes.
Maybe while the students are getting their thoughts together and putting up questions, just add a few points to what Susan mentioned. Is it okay, Susan?
Yeah,
While we are waiting? Yeah.
actually, you will be knowing better than me.
No, no, not that way. I just thought I'm just, instead of just waiting, mum. All the students must actually, after they've written their paper, they must reread the paper And it's very, very important to reread the paper that they have written, so that the content of the introduction is actually now reflected in what your results have come and what is the discussion showing. So sometimes you may need to add something more to the introduction in case your results, show some interesting points which you had not thought about when you started, and that's very important. Second point is that you should also be looking at objectives, whether your objectives have been met from the results. So when you read this paper over again, it should be very clearly evident that yes, your results have met the objectives of the paper. Another one which I believe very, very important, I think very nicely Susan has mentioned about plagiarism, very important factor these days. The other thing is that whoever is a co-author, you must have them read the paper and they must sign the that they have read the paper. It's very important these days. Uh, there should not be any gift authorship. You should not say, I'm a friend of yours, so please put my name on the paper. These are points which come up, and you may think about them. And I mention this because it is happening, and it is important to note that where you are actually writing who's doing what part of the paper, it really clearly defines what you're doing. But sometimes some papers may not ask you for that information. Then you need to be a little cautious. So do ask your co-authors to read the paper and also sign that they accept what they read, or give you suggestions for making any changes in the if they don't agree with you. And in this process, there's a nice internal review of your own paper. It's a very good way of functioning, and it helps you to improve the quality of your paper. So do think about all of this while you're doing this. Paper writing is now very important, and people are very, very cautious. And as Susan rightly pointed out, there are ways and methods today to find out where you have fudged pictures. And there is a system called as PubPeer, where if you are you are saying that adding a particular reagent, kill the bacteria or reduce the number of bacteria or increase the growth of the cells, and you show pictures with this information, they can make out whether they are fudged data. So be conscious about all of this, and please do not fudge data. That is very important. It can be caught when it is being reviewed. So be very cautious about this. So Shweta, leave the platform. I leave it for the youngsters to
Um,
ask questions.
Ma'am, there is one question we have received. Kindly explain H index.
You take that thing. H index is something which made by Hirsch. It quantifies the actual scientific productivity of the apparent scientific impact of a scientist. It is suggested by Hirsch. Suppose you have a like a H index of you have 25 papers, each of which is cited 25 times, then H index is 25. You have some paper which are cited, one is cited 10 times, one is cited 9 times, one is cited 5 times, one is cited just one time, then H index will go down.
Thank you, ma'am. As we end the
You
session,
can go
I
to
would like to
if you
thank
have
Dr. Mala Kharkar.
Google
Yes, ma'am.
scholar.google.com, and then you can find out if you have published some papers, what is your H index. You put your email ID and some details, your institute name. You can find your H index.
Okay, we
H
have
index
got
is
one more question.
okay.
What specific actions of the author is considered plagiarism?
What?
What
Can you
specific actions what specific actions of the author is considered plagiarism?
Plagiarism is something in which you are taking cut and paste from somebody's article, either a figure or a table or a text, without giving the actual reference. If you give the reference, absolutely no problem. But if you are just cutting and pasting, nowadays students are just cutting and pasting from somewhere and putting it into their thesis and all that. That is plagiarism without giving credit to that earlier publication. So our students actually, they really don't know about much about plagiarism, and they just cut and paste. They're very good in that, without giving credit. If you give credit, absolutely no problem. And the guide will not know actually, unless he or she checks, how the student has cut and pasted things without giving reference. Then only the guide will come to know. And if such a paper is published, now maybe there are softwares, but earlier they used to be published, and those papers are retracted. It's such a bad thing for impact for the institute also. So students have to be very careful, regarding the writing things from other things, copying things, and reproducing things without reference. If you give the reference, but if you want to reproduce a table, you have to take permission from that particular author.
Thank you, ma'am. Any further questions from the participants? Okay, I guess we shall end the session now.
Yes, please. Yeah.
We have received actually one more question. Madam, can you please let us know where can we detect plagiarism? Is there any free tool on the web?
Yeah, there is a software, I think from the library. I'm not very familiar with that software to check plagiarism. 1 or 2% is allowed, not more than that. And all the journals also have that software to check, and big institutes are having. I don't know about the universities. Maybe somebody from a college or maybe can give more details. I don't know about whether Sureka knows about this software.
You can actually, if you just say check for plagiarism, you can see it. They may not be the very best of. Um, if you in Google, you just write check for plagiarism in your text, you will get some software which can be used, and that will be a good first starter. I'm sure there will be much higher end versions of the software, but do try that as a first starter for yourself.
The next question is copyright photographed can be used mentioning the source.
Yeah, but you have to take permission from the author. In your research paper, you don't have to copyright from somebody else. Only if it's a review article, you can reproduce some papers from somebody else, some other published paper. There, you have to take a permission from the author or from the journal or whoever, wherever it is, and then only use it for the review paper. But in a research paper, there is normally no need.
Okay,
While
any
you're
more
sitting
questions?
there, the youngsters can actually type in scan my paper. You will find at least 4 or 5 different software for checking this. It's easier to do it. Yeah, and Suparna has already put it in, and I think here's one more which I
There is.
saw. So I've just put check my paper for plagiarism in Google, and you'll find these.
Okay.
Okay, and that should be a good starter point for you. But But as mentioned, copying of tables is not permitted. If you do need to, you have to take permission not only from the author but from the publisher. And in your thesis in particular, if you're just taking a picture without changing it, you really have to take not do that at all. Even I understand recently that you cannot even say that you adapted it from somewhere else. These are the recent ones that I learned a few months back. I thought you could just give the reference. So things are changing very rapidly. So you need to check these out before just taking a picture and pasting it. Very often we tend to take ideas of how a figure is to be made and then change the figures to suit your requirements. Maybe that could be done, but it should not look like the original one at all.
I guess there are no more questions from the participants. So now, as we end the session, I would like to thank Dr. Mala Kharkar, CEO of Patkar-Varde College, Chief Coordinator Mrs. Ruchita Dalvi ma'am, and Co-Coordinator Mrs. Anupama Tomar and Mrs. Neha Kolako for their constant support. I wholeheartedly thank Dr. Susan Nippen ma'am for taking out time from busy schedule and delivering such a wonderful session. Lastly, a big thank you to all the participants for participating in the webinar. The feedback link has been posted in the chat box. You can fill up the link to receive the certificates. Thank you, Vex. Thank you, all of you.
Thank you, Susan, and thank you everybody at Patkar College. I have to mute myself because there's some work going on around here. Makes a lot of
Okay.
noise.
Okay. Bye
So
bye.
pardon me for that. I will disconnect now. Thank
Yeah.
you.